Okay, let's dive into this. Welcome to today's explainer, where we are going to unpack one of the absolutely most fascinating areas of biotechnology out there, plant tissue culture. We're going to explore exactly how scientists are taking biology that happens naturally anyway and literally supercharging it in the laboratory to solve some of the world's absolute biggest agricultural challenges. But right off the bat, I want you to think about this question. What if you could grow an entire forest from just a single tiny leaf? I mean, it sounds like pure science fiction, doesn't it? But what if I told you that this isn't fiction at all? It's actual, established science. Today, we're looking at the exact mechanics of how that works and why it's completely revolutionizing the way we grow everything from the food on our plates to the medicine we take. So here is our roadmap for today. We're going to cover 1. The power of one cell. 2. The tissue culture recipe. 3. From cell to plant. 4. Real-world applications. And finally, 5. The future of farming. We'll journey from the microscopic magic of a single cell all the way to global agricultural solutions.
All right, Section 1, the power of one cell.
The foundational secret to all of this basically comes down to one incredibly powerful word, and that is totipotency. Totipotency is the ability of one single plant cell to divide, differentiate, and ultimately regenerate into a complete, fully functioning plant. Just imagine for a second if you had the superpower to regenerate an entire perfect human being from just one scraped skin cell. That is exactly what plant cells can do under the right conditions. The process involves cell division, differentiation into specialized tissues, organ formation, and then boom, total plant regeneration.
Moving right along to Section 2, the tissue culture recipe. Let's transition into the laboratory. Think of this kind of like a high-tech cooking class. To bake this particular scientific cake, you need a few highly specific ingredients. First up, the explant. This is your starting piece of plant material, which could be a leaf, a stem, a root, or even an embryo. Next, you need the broth. The most common one is the Murashig and Skug, or MS medium, which provides all the vital macronutrients, micronutrients, vitamins, and sugar, sugar, that the plant needs to grow. Then you add agar to solidify it into a gel. But here is the absolutely critical part. Everything must be done under perfect sterile conditions. We're talking autoclaves to sterilize equipment and laminar airflow cabinets to filter the air. Because if even a single stray bacteria or fungal spore gets in there, it will feast on that sugar in the medium and completely ruin your whole culture.
And this brilliantly illustrates our chef's most special ingredients, plant growth regulators, or hormones. These chemical messengers dictate exactly what our plant cell is going to become. On one side, we have auxins like IA or NAA, which promote root formation and cell division. On the other side, we have cytokinins, which promote shoot formation. By just tweaking the ratio of these two hormones in the nutrient medium, scientists can literally command the plant cells to either grow roots downwards or shoot leaves upwards.
Okay, Section 3, from cell to plant. So, there are 6 methodical steps here in the lab. First, we select healthy plant material and surface sterilize it with chemicals like sodium hypochlorite or ethanol to wipe out any surface microorganisms. Step 2 is inoculation, where we carefully place this sterilized explant onto our nutrient medium. Step 3 is callus formation. Step 4 is organogenesis or embryogenesis, where the actual shoots and roots start to form. Step 5 is full plant regeneration. And finally, Step 6, acclimatization. I actually always like to think of this step as a plant moving out of its cozy, highly pampered, sterile basement laboratory and trying to adapt to the harsh reality of real-world soil and weather. It definitely has to happen gradually. Now, I want to pause briefly on Step 3, actually, because it is so unique. A callus is simply a mass of unorganized, undifferentiated plant cells. Think of it as a blob of very confused stem cells just sitting around on the agar, waiting for the chemical instructions, those auxins and cytokinins we just talked about, to tell it what to do next. It is literally the ultimate blank canvas of the plant world, and it's heavily used in genetic studies and secondary metabolite production.
Section 4, real-world applications.
So the crucial point here is that this isn't just theoretical science confined to a lab. It's practically sitting on your dinner plate right now. One major application is micropropagation, which is the large-scale production of identical plants. If you've ever enjoyed a perfectly uniform banana, potato, or strawberry at the grocery store year-round, you are very likely benefiting from tissue culture. Beyond food, meristem culture is used to produce entirely virus-free crops like sugarcane. We use these methods to conserve highly endangered, rare plant species by preserving their germplasm.
We even use cell suspension cultures to mass-produce valuable secondary metabolites, like alkaloids for pharmaceuticals, flavonoids, and essential oils, all without having to harvest massive amounts of wild plants.
To put that into perspective, the scale of this efficiency is truly mind-boggling. Through micropropagation, a single tiny explant in a lab can rapidly multiply into thousands upon thousands of genetically identical plants. Because it requires such small plant material and relies on controlled indoor conditions, this massive production can happen year-round, completely independent of the weather or the seasons outside.
Which brings us to Section 5, the future of farming.
By combining the fundamental tissue culture techniques we've discussed with modern genetic engineering, scientists are essentially creating superhero crops. We're talking about deep genetic transformation and mutagenesis to design plants with improved nutritional traits and incredibly powerful pest resistance. This allows us to select and scale the absolute most resilient varieties, paving the way for the sustainable, high-yield agriculture we need to feed a rapidly growing planet.
Let's move to and see how this builds into a quick recap. We can map out exactly how specific culture techniques match up with their world-changing applications. Micropropagation gives us rapid plant multiplication. Meristem culture guarantees virus-free plants. Anther culture, which uses pollen-producing tissues, allows breeders to produce haploid plants for much faster pure line development. And protoplast culture, where scientists work with plant cells that have had their cell walls removed, well, that opens the door to deep genetic transformation and somatic hybridization. It is an incredibly versatile toolkit.
So, I will leave you with this provocative thought to ponder. As our climate shifts and our global population continues to grow, these sterile lab techniques are stepping out of the shadows and into the sunlight to ensure our agricultural survival. How will plant tissue culture shape the food on your plate in 2050? Will your next meal be a climate-resistant, nutrient-boosted masterpiece born from a single totipotent cell? Thank you for exploring the incredible science of plant tissue culture with me today. Keep asking questions and keep learning.